Hi, my name is Surush Barsa, and I'm an entomologist at CIAT, the International Center for Tropical Agriculture. The overall objective of this video is to demonstrate a method to characterize mechanisms of host plant resistance to herbivores in support of crop breeding efforts.
Our breeding initiative to enhance spittlebug resistance in Brachiaria grasses will provide an example. Spittlebugs are serious pests of many economically important grasses, including tropical pastures, sugarcane, and corn. In the American tropics, pastures belonging to the genus Brachiaria are their most important host. A plant can resist herbivore damage through three broad mechanisms: antizoonosis, antibiosis, and tolerance. Antizoonosis, sometimes called non-preference, is the degree to which the plant is avoided when the herbivore is able to select other plants. Antizoonosis is typically detected in choice tests, where herbivores are given access to more than one genotype. Antibiosis is the degree to which the plant affects the fitness of the herbivore feeding on it. Finally, tolerance is the degree to which the plant can withstand or repair damage caused by the herbivore without compromising the herbivore's growth and reproduction. Both antibiosis and tolerance are best detected in no-choice tests, where herbivores are confined and therefore forced to feed on a single plant. Our video will demonstrate a no-choice test designed to estimate antibiosis and tolerance to spittlebug herbivory in Brachiaria grasses. Host plant resistance is a key spittlebug management tactic. However, resistance based on strong antibiosis could create selection pressures for more virulent pest biotypes. On the other hand, resistance based on tolerance would allow spittlebug populations to continue growing until the numbers overwhelm the plant. Durable resistance, therefore, may require some levels of both antibiosis and of tolerance. This protocol will demonstrate how to estimate the relative contributions of each to spittlebug resistance in Brachiaria. Brachiaria genotypes are obtained from the germplasm bank of the International Center for Tropical Agriculture. The plants are vegetatively propagated from single-stem cuttings from mature plants used as experimental units. The cuttings are trimmed to 10 centimeters to ensure uniformity of planting material. To prevent contamination by plant pathogens, the cuttings are washed in a 3% sodium hypochlorite solution for 2 minutes and then rinsed thoroughly with tap water. Each cutting is planted in approximately 36 grams of sterile soil in a PVC tube sealed with the lower section of a styrofoam cup and capped with a PVC bushing. The cutting is held in place by a foam ring placed in the central opening of the bushing. Plants are fertilized and water is needed to maintain adequate soil moisture. The plants will grow for 1 month before being challenged by insects, allowing development of superficial roots, which serve as feeding sites for root-feeding spittlebug nymphs. Inverting the plants for 8 days prior to infestation further stimulates the growth of superficial roots, partially adjusting for inherent differences in root architecture. This step requires an artificial source of light, which we provide for 24 hours a day. Plants with little or no superficial roots after this inversion treatment are eliminated from the experiment. 30 days after planting, each plant is infested at the soil surface with 6 mature eggs obtained from our spittlebug colonies. To establish these colonies, adult spittlebugs are collected from the field, identified as species, and introduced to oviposition cages where they are provided foliage of susceptible Brachiaria to feed on. Females lay their eggs in soil contained by a shallow, removable tray placed on the bottom of the cages. After several days, the soil is suspended in water and passed through a series of sieves to collect the eggs. Suspending the sieve material in a 30% saline solution causes the mature eggs to float, leaving behind the mature eggs and any residual organic matter. To prevent contamination by insect pathogens, eggs are disinfected in a 0.5% sodium hypochlorite solution for 5 minutes and then rinsed with distilled water. Only mature eggs close to hatching are selected for infestations. They can be identified by the presence of 2 red dots in the anterior part corresponding to the insect's eyes, pink marks in the posterior part corresponding to the insect's abdomen, and a fully expanded operculum. Successful hatching is confirmed 4 days later, at which point any unhatched egg is replaced by a neonate spittlebug from our colony. Test plants are arranged in a randomized complete block design with 6 replications. The experiment is evaluated approximately 30 days after infestation. Scoring for plant damage provides an estimate of tolerance, while scoring for insect survival provides an estimate of antibiosis. Damage is scored using a 1 to 5 visual scale, where 1 corresponds to no visible damage and 5 corresponds to plant death. In turn, survival is assessed by counting the insects reaching either their final insert or their adult stage. To categorize resistance mechanisms, it is useful to visualize results using a biplot, where insect survival is on the x-axis and plant damage on the y-axis. Antibiosis increases as one moves from right to left. On the other hand, tolerance increases as one moves from top to bottom. We consider plants resistant if the response to herbivory falls in the bottom half of this plot. If this response falls on the left quadrant, the dominant resistance mechanism is antibiosis. If the response falls on the right quadrant, the dominant resistance mechanism is tolerance. Here's a representative sample of our screening data. The points on the top represent susceptible plants. The points on the lower left quadrant represent plants resistant by antibiosis. The points on the lower right quadrant represent plants resistant by tolerance.
We've just watched a demonstration of a no-choice assay to estimate the relative contributions of antibiosis and tolerance to plant resistance to herbivores. We use spittlebug resistance in Brachiaria as an example, but the principles presented should be relevant to other systems. We hope our video helps you in your own efforts. Good luck!